Good afternoon for everyone participating in this very exciting webinar. The title of it, The Challenges of the Medical Device Innovation Ecosystem. This webinar is co-sponsored by the International Society for Cardiovascular Translational Research, ISCTR, and the University of Arizona Health Law and Policy Program. As you're all aware, that the innovation system is very complex system, and currently we spend about 15 years and $1 to $2 billion to develop a medical device or therapy. And that's because of the complexity, as you see here, from the finding the unmet need to the concept, the prototyping, the preclinical evaluation, the clinical evaluation, regulatory and reimbursement strategy, practice guideline strategy. This is unmet educational need. We do not learn this in our school, whether engineer or physician and so forth. So as you see here, the complexity of this system resulted in 1 out of 3,000 idea, and less than 10% of startup company will succeed, will reach the concept, will reach a useful product. Our mission in the ISCTR is to address the challenge of product development and to standardize the methods and endpoint for a preclinical and a clinical medical device development. What we are calling today is the translational pathways for medical and cardiovascular devices. We were able to, over the last several years, to collaborate with over 500 experts from a different background, academia, industry, regulatory, reimbursement, guideline, and patient, to put together a platform for medical device evaluation. And currently we have online course with a 36 lecture for the general medical device evaluation, and another online course of 80 lectures on cardiovascular devices development. In addition to that, we do have a reference of a document of 400 pages to support and expand on that online course. Also, I'm happy to say that over the year, we have a unique educational program in the translational medicine. And we do have also a journal that established in 2008 and very successful, distributed by Springer Nature worldwide. As a result of putting this a unique educational format, basically we are trying to put this translational education in a formal education. We are now collaborating with many universities to add to their education program this specific type of education on medical device development. And today is we have the James E. Rogers College of Law that will be collaborating with ISCTR to put a unique program for their students. Professor Tara Sklar, the Professor of Health Law and Director of Health Law and Policy Program at the University of Arizona, very soon will shed some light on that unique program. We are very excited and thankful for our speaker today, keynote speaker, Dr. John Simpson. John Simpson's innovation has impacted the life of so many patients worldwide. And he will be introduced by Professor Spencer King, Emeritus Professor at the Emory University, and he's a Chairman of Education of the ISCTR. And with us also as a panelist, and Dr. Anthony DiMaria, who's a Professor of Medicine at UCSD and Vice President of ISCTR. So Tara, so joyful to work with you on this unique program. And please go ahead and tell us about the program that a result of the collaboration between the society and the University of Arizona.
Sure, thank you so much, Dr. Dib. The feeling is absolutely mutual. It is such a pleasure to work with you and ISCTR. We're delighted to co-host today's webinar. We also are thrilled about the course that will be starting in just a few weeks, led by Dr. Nabil Dib, called Translational Pathways for Medical Devices. It's an online 6-week course, and many of the issues that you'll hear about today will have a much more in-depth dive in that course. You may be wondering how a College of Law got involved among the other engineering and medical and science programs. As many of you know, in practice, legal issues are constant in medical product development, especially with medical devices. We hope, and quite often we have folks that have no real interest in practicing law for a myriad of reasons, but they still want to be able to identify and resolve legal issues, many of which can help navigate those common barriers that lead to such a high rate of failure among the startup companies. The new course we plan to offer will run annually every summer. It's part of our graduate certificate in regulatory science, as well as our Master's of Legal Studies in Health Law and Policy. What we really are trying to do with this course and program is fill a lagging gap in higher education, where most institutions, the sciences, engineering, law, and business are separate. I'm happy and delighted that most of my students are actually not JDs or lawyers. They are researchers, clinicians, engineers, entrepreneurs, like many of you in the audience. We actually have over 100 individuals at RSVP today from all over the world. And in addition to that, we have a number of students that are part of our regulatory science program, who I'm sure are taking studious notes because this webinar is part of their course and they will be graded. With that in mind, I'd like to note that Professor Karen Goff will be monitoring the Q&A. So please, if you have any questions at any point during the webinar today, to submit them in the Q&A, and Professor Goff will be sure to lead them to our keynote speaker, Dr. Simpson, and our panelists at the end of near the end of the hour. And I also just like to add that we hope this is the first and the starting point for future events with ISCTR, and we really encourage you to email us with any ideas for future topics going forward. So thank you so much for joining us. I'll turn it over to Dr. Spencer King, Chair of the ISCTR Committee.
Well, thank you very much. Yes, this is all about unmet clinical needs, as Nabil Dib has said, and we have with us as our speaker someone who has committed his whole life to unmet clinical needs. And in this case, they're mostly within the arteries of our bodies. They've got to do with devices to open arteries, to view what's going on inside the arteries. It's almost like the old movie Fantastic Voyage. John Simpson is maybe the Raquel Welch of swimming in the arterial system. John was a Texan at one time when he started out and went to Ohio State to college, went to Duke for medical school, and then did a PhD back in Texas at the university, and was in immunology of all things. It could solve some of our unmet clinical needs in immunology right now. But then he went to Stanford, and while at Stanford, got exposed to a fellow that had invented a balloon to open up coronary arteries, Andreas Grunzig. And so John took interest in this, and I'll tell you just a short story. I met him first in Zurich in a bar, actually an outside bar. And John was interested in taking this idea that he had about developing a unique kind of system for opening coronary arteries. And he asked me the question. He said, should I start a company or should I sell this idea? I'm exposed to my own acumen in this area is exposed by the fact that I told him that if somebody was going to offer him actual cash money for this idea, he should take it and run. He didn't. He started his first company, and the rest is history. John has started about 6 or 7 different companies that have been pivotal in changing what we do in cardiology and interventional cardiology. And John, I look forward to having you share some of the ideas with us today. You're muted still. There you go.
Okay, now can you hear me now?
You're good.
Good. Yeah, thanks, Spencer. I remember, I think we were having mimosas out on the deck. So it wasn't, and it was outside dining, so would have been okay for COVID.
Would have been appropriate.
Even in this era, I would say. Well, this is really a privilege for me today. I'm just learning that I'm speaking to a large number of lawyers or lawyers to be. I have had some experience speaking to a large number of lawyers in the past, mostly favorable, but I'm hoping that these are all favorable. That would be definitely would be exciting. Mostly students, and I just absolutely feel like this is a special privilege to be speaking to students. This has been my passion. It was Andreas Grunzig's passion in the very early days. I think I tried to pattern a lot of what I've done after Andreas Grunzig. He was absolutely an extraordinary model for me, as he was for Spencer, I would say. So I'm going to share my screen. Can I do that now? Yes.
Please.
Okay, let's see if this is going to work. All right, good. We're going to talk about a Nuco today. How does it take to start a Nuco, as in a new company? And if I can get organized here, then that's the last slide in my presentation. This is my first slide. So this is about Andreas Grunzig. And Andreas Grunzig, as Spencer's already alluded to, was the father and the founder of this. And throughout the centuries, according to Ayn Rand, there have been men who took their first steps down new roads armed with nothing but their own vision. What a perfect way to characterize Andreas Grunzig. I would say that there's no better way to say he ventured off on this task all on his own to really change in a dramatic way the way vascular disease was treated, and his innovation and let's say leadership and motivation to all the rest of us has been pivotal to seeing the way vascular disease is currently treated in the United States is totally different than what we might have imagined prior to Grunzig's involvement in this. So I wanted to introduce Andreas, and some of you may know him, but Spencer was a very close colleague with Andreas, working with him for many years at Emory University, as well as Anthony DeMaria. I knew him very well. So this is he's a special person. Here are two wannabes, Andreas Grunzig. This is me, and this is Richard Myler. This is Andreas Grunzig, and we did have some wine that night. There's a cork around here somewhere where you'll see more of that later on. But the two wannabes primarily at that point were me and Richard. Wanted to be like him, but we also wanted to share the wine, which is also really very good. This is the very first catheter this is the first catheter that I ever made back in 1978 in Menlo Park, California. And it's a plastic tube that allows a guide wire to go down the center of this plastic tube. It has a balloon on it, and it allows you to go in and to treat coronary arteries in a way that was unique at the time, because the previous catheters had a so-called fixed wire system down on the tip. I'm showing this mostly to tell you how, I don't know, how bizarre the start always is, and the way how ugly it is. You have to have real confidence. And what I would say to the students, your confidence has to be only focused on one thing, and that's making things better for the patient. If your strategy is to how can I make so much money you can't stand it, or how can I exploit the system, or how can I beat the regulatory process, or no, any of those things. So none of that stuff. I mean, I guess people have made it occasionally work. Rarely does it work, and I really oppose that as a motivation. Stay motivated on making things better for the patient, not just making the device a little bit more streamlined or a little different color or anything along. What is it going to take to get a better outcome for the patient? So as it relates to this particular device, I tested this device. You can read in the text here. I unfortunately got a NASA baboon that had 40-pack year smoking history and ate mostly peanut butter. Tried to see if we could open up their coronary arteries, assuming that baboon had a narrowed coronary artery. Turns out he did not. But he did fibrillate during my less sophisticated than I had hoped, I suppose, skill sets at NASA back in that era. So the guiding catheter got sucked down into the coronary artery. The baboon fibrillated, and the defibrillator would not work. It was about the size maybe for a small dog, and it did not work on this 400-pound animal. NASA terminated all my future studies. I never got a chance to really test the balloon any further in the primate model. The point here, though, being that when you start to work on the development path, and it's very different now. I mean, honestly, the first device that we used in patients, the only thing that was very different was that the adapters were no longer braised bronze; they were plastic. Beyond that, everything else was pretty much kind of the same, and we could actually test that in patients back in that era. As Spencer will tell you, that most of the early tests were not so favorable in most settings, I would have to confess. So let's talk about Nuco and what we're going to. How would we approach starting a new company in a more modern era, let's say today? And let's say we're going to look for an image-guided reentry mechanism. The way that we would traditionally do this is that we would go out and we would make sure that we have a real unmet clinical need, which is my obsession. The unmet clinical need here is a coronary artery when the catheter is in the so-called subintimal space and has to get back into the true lumen. When that's the case, I've got something beeping in the background. I apologize for that. But I'll just kind of go through this, and these are all things that you already know about. That we need to, you always do a mission statement, which I think is a waste of time, but it's required. You put together the management team. That's the key, as far as I'm concerned. Look at the technologies and what kind of innovation is it going to take to execute on the plan that you have identified to meet the unmet clinical needs. Always focus back on the unmet clinical need and what will it take to do that. Then, of course, then you have to look at the market size to attract the dollars that you need to finance the plan. And then everybody wants to ask about an exit strategy. I can tell you right now, there's no way to predict the exit strategy for any of these startup companies. You sell it or an IPO. Okay, those are the most traditional. Other kind of partnerships are possible. So it's really hard to predict. I just point out, I did not prepare this slide, so it looks like I'll be bragging about all the stuff that I've done. But just to say, we have two, say in this particular theoretical company, we might have two founders. Suppose I'm one founder, and I've had this experience with Fox Hollow, Preclinical, CVS, DVI, ACS. Goes back. So we've raised a fair amount of money. Like you say, we raised a fair amount of money. We spent a fair amount of money. And we've sold these companies for also a fair amount of money. We have also a technical founder, which I think is pretty common. I would say that in my experience so far today, the first thing that I do is to find an engineer that knows a whole lot more about how to do what I need to do than I do, and allow that engineer to perfect this. Now, let's say in this case, maybe we're going to look at some kind of an imaging system. We need somebody with experience in lasers and along those lines. So we might seek out that person just to show you they can really work. And this is the only slide that I will show you that will have any financial data on it. But these are the honest and true daily sales numbers for Fox Hollow, which was a company that we started with the unmet clinical need to treat peripheral artery disease. It's not for use in the coronary arteries. But at one time, we averaged, with this regression model, you can see we're averaging about $700,000 a day in sales. And so those are generous sales. And then this company eventually sold for $740 million, so about 1,000 times daily sales, if you will. Most of these companies will sell for anywhere from 5 to 10 times sales on an annual basis. So this is kind of a funny number that just turned out to be more of a coincidence, I think, than anything else. I just wanted you to see that because it could actually work. Of course, I tell everybody I did all this by myself. That's a slight exaggeration. Eventually this company had 200 employees. I'm sorry, they had 200 salespeople. and again, close to 300 employees. So it's a huge team effort. And that's the key is finding, becoming obsessed about a better clinical outcome, and then putting together the team that can execute on that. That's really the key. And quite frankly, most of them are a lot younger than I am, but it's thrilling to work with them. The basic premise of what this new company, let's say, would be doing if they're trying to use imaging is that, seeing what you're doing, and while you're doing it, can make it a better outcome. How you do this is the rocket science. The concept is not that you see what you do is not rocket science. How you do it is, and maybe you need a real expert that could help put that all together. So the Nuco then is committed to meeting these, this is a little bit redundant now, but unmet clinical needs are really, really the key. And particularly in the cardiovascular disease space, being able to see inside the artery while you're working on it. And maybe in this example, perhaps we would use OCT, optical coherence tomography, as a technique for looking inside the artery, looking at the artery wall. And maybe we'd even use AI to help direct the device, and then might even use robotics. And these are buzzwords, but I think they're important if you can actually explore them and bring them to bear in a way that's useful and that facilitates a better outcome for the patient. These are just all, I'm gonna skip this slide from a different presentation, but just to show you that, suppose that we would look at what happens if you try to cross a normal, a totally blocked coronary artery. You might put a loop guide wire down in this area. It's called antegrade dissection and reentry is the term for it. Spencer knows it well as Anthony, and you put the guide wire down, you form a loop in the guide wire, and you go down and you dissect the artery. See the guide wire with the loop on it, you go down and you dissect the artery. And then you put in some kind of a catheter. There are many catheters that are available there. This is the only one that we believe has OCT on board. And as we put this catheter down in the suboptimal space, we can have an imager mounted inside of it. And the imager would allow us to orient this device in a way so that we could put a guide wire back into the true lumen. This is our target. We want to aim over here. This is a lot more detail than you ever really want. But just to say that in this setting, the unmet clinical need is, how do you get out of the bad track into the good track? And can imaging help you get out of the bad track into the good track? And the answer is yes, that it can. And we've shown in experimental models that would be possible. Now, I'm going to skip this slide. This is just an example of a human coronary cadaver vessel that we're able to show that this actually works. We can get this guide wire back into the correct artery. But I think this is, based on the audience, that would probably be more than anybody could tolerate at this point. And then, you always have preclinical milestones, and that was outlined in some of Dr. Deb's earlier slides. But to me, the preclinical milestones are really kind of key, because they would help you say, are we able to address the clinical needs that the patient really can benefit from? So in the preclinical testing, if you find out that this device does not work in a way that is going to yield better patient outcomes, for example, a balloon that is, let's just say, in the era who are just making balloons that had a guide wire that you could direct, if being able to direct the guide wire and aim it and go places didn't benefit you, then that device is not going to have much benefit. If, on the other hand, being able to direct the guide wire and put it in unique places allows you to access arteries in a unique way that provide clinical benefit for the patient, then that's a genuine opportunity. So, and the investment opportunities, vary a lot. I just put these numbers up because these are numbers that would say for a small startup would not be so unusual. Frequently, the financings are done now with convertible notes. You might put in $2 or $3 million in the first round. $10 to $15 million valuations on some of these technologies coming out of a Series A round. Let's say a second round might target a $20 million pre-money valuation. Those are just kind of general targets that are pretty common in the space. You hope to sell these companies if you're looking just at the finances of it all, and you're ignoring the patient opportunities, which I advise against. you still would expect to sell the companies for a nice multiple from a business perspective of what you have invested in them. So I do want you to know, though, that I was at that meeting with Grunzig, and I did get this cork, and Spencer has seen a picture of this cork 1,000 times, but you can see that we signed the cork that night back in '78, and we gave ourselves some appointed positions. This is AG signed the top of the cork, and he underlined that as he is chairman of the board. And then Myler was something else. He was marketing or forgot. Anyway, they did not know what to do with me as a cardiology fellow at Stanford at the time. I'm JBS though, and you can see the cork is pretty torn up, but this is the bottom of the cork. This is the top of the cork where Andreas went in. I'm the bottom of the cork, and my job was just to keep the cork. I'm the KC. So this confirms that I've kept the cork long enough to get a photograph of it. I can't say I could produce the cork today, but I can produce these photographs which document what it is. Now, the next slide is a greeting from one of Spencer's a young woman who idolized Spencer King, and I talked to her last night, and she told me to tell Spencer hello. She misses seeing him in Hawaii. So this is my daughter that had a cerebral hemorrhage. What's been 4 or 5 years ago. That's when, after she had the hemorrhage, and it's been fixed, and here she is today. 0 residual, and she told me last night to tell Spencer King that she said hello. So Spencer King from Kendall, Rolene, Simpson, Kendall, Simpson, Rolene, now hello. And she said she just did Mauna Kea last week, and it looks exactly the same as it did when we went there for your symposia in the past. So with that, I want to say from this our Nuco experience here, thank you very much.
John, thank you so much. I appreciate the shout out, and I missed those days at Mauna Kea. But when you talk about starting these new companies, and when you started your first company, the things that are obvious to me that you did was to figure out what you could do and what you needed help with. And you got Ray Williams to raise you a lot of money, and you got engineers to build your devices and all that. And I think everybody can kind of conceptualize that you've got to have these ingredients. Where along the way, I'm thinking we got all these lawyers listening in, where along the way do you delve into the IP and see if you've got something? Do you worry about that when you know you've got an unmet clinical need? Do you sort of say, well, I would go ahead and develop this, but maybe it's too much trouble. Maybe it's all been done already. Maybe somebody's got this covered in a broad patent. When do you worry about the IP?
Mm-hmm. Yeah, that's always a good question. I think you worry about the IP always. You never ignore the IP, but I don't think it's ever the focus. I know Paul Yock and I differ dramatically on our focus on this, and we feel strongly that he feels strongly that you really focus on IP from the very first day. I say, you've got to make some prototypes. Let's see if this works in some kind of a model. You have to have some confidence that this is going to be real beyond just your fantasy that you need to file a patent. And if you can get a patent filed really quickly, then that protects your space. You should file patents. There's no doubt about that. And there should be some protection, not the focus though. I don't think that is really the correct strategy, but I think I may be in the minority on that.
Well, there are ways to do things, and you've done them the right way, and the ways not to do them. And Andreas Grunzig and I would say, probably have fit into that category, because Andreas, who developed the balloon originally, had a U.S. issued patent on balloon dilatation of arteries, including the what we'd have now with stents and the rest of that. But he had published on this well before he applied for that patent. So the patent was, of course, invalidated because of his not knowing you had to do that back in those days. So this IP is critical in developing anything.
Spencer, could I ask John a question?
Absolutely.
So, John,
this is Dr. DeMaria, by the way, who's
hi, Anthony. How are you?
At least fair to middling, but in any event, My question goes back a little bit to the IP, because as scientists, often what we want to do is get our paper accepted at a national meeting and get up and present the data. And how does that influence the IP?
Yeah, I think you could do, this is one thing I think you could actually do both. You're going to want to get, before you do the presentation, any presentation that we would be giving, as in the one that I'm giving to you today, has been reviewed by an attorney. And so the attorney has bought off on some of it. I see now that I've got a mistake on this slide that I've got up here still, which the attorney would be miffed if he saw that. So you guys just need to ignore it. But yeah, you have to pay really careful attention to this in this modern world. I would say maybe in the world that I first got started in, and with what Spencer was alluding to, the IP as related to the Grunzig patent and my patent. I had to be honest with you. I told my patent attorney, I said, "There's no way that this is patentable because Andres has already done it." And he said, "Well, let's look and see what Andres has done." And so we did, and I don't know, according to them, they got a patent on it. I don't know how that all kind of worked out, but the legal aspects of this are really critical. They're really, really, really important. They cannot be the focus, is what I would argue. And you can for sure give presentations, and you can have them screened in advance of the presentation to see if you're disclosing anything that's IP sensitive. And you can definitely be covered in that space.
I'd just like to chime in real quick, if I could, from a legal perspective. I just thought the court was such a great example of the other ways that legal issues get involved right away, which is really contractual, governance structure, and other things beyond just IP. And I actually I've seen cocktail napkins with photographs on them, but never a court. That was a first. And also, if you don't mind, Dr. Simpson, could you unshare your slides so that we can all see each other again?
Yeah, I would like to unshare them. Actually, I was trying to do that, but for other reasons. But stop sharing. Yes, please. Yes, that's better. I like that better too.
John, you've been so effective in building a team around you for all these kind of things, and it occurs to me that these lawyers or law students who may not practice law, as you indicated, they might find a better thing to do, are unlikely to be the people who recognize the unmet clinical need. So the teaming up of people who are experienced in business and law and in fundraising and so forth, how do you build a team? How do you get the people together that you need to do that? Even though you might have come up with the idea, did you recognize early on that you needed help?
Oh, Spencer, I'm so smart. I don't really need any help. You know, I'm able to do all this stuff by myself. I'm so amazing.
It's a rhetorical question.
So I mean, help is, absolutely. And so here's the usual sequence, which I didn't allude to. I don't think I didn't very well characterize exactly what we do. The sequence almost always is come with the idea, find the engineers to make it a reality. My ideas are frequently, so my best ideas could never be reduced to manufacture. I've got one idea that nobody can make it, but boy, if they ever could, it would be really cool. So always trying to get the idea, they get somebody who can make it, make some prototypes, and they get the lawyers to put together the structure of the company. So lawyers help us more, I would say, in this phase. They're more helpful in how you structure the company. You know, you have to structure the stock option programs. You have to, put together the investment documents for even the early round Series A, even his family and friends. You still have to have documents. That's very important. And so right away, maybe I didn't allude to that. Right away, you get the legal side of this gets involved. I'm thinking about this sort of our recent efforts right now. We had our attorney. I guess he's probably he's not on full time, but he's probably our fourth person that we reached out to beyond one. So it's me, two other engineers, then the attorney.
I've heard you somewhere pontificate a little bit about some of these other ingredients that Nabil has shown us that you need going up that pathway. And one of them is FDA, another one is CMS, or, so regulatory and reimbursement. My impression is that you don't think that's such a difficult milestone in your process. Is that right?
Well, no, I do think they're kind of hard. They can be hard and kind of tricky, and how you navigate them, I think now probably requires more sophisticated effort than I might have thought at this phase. But I think, again, as long as you're honest with the FDA, and I think you and I, and I think all of our colleagues have tried to be as honest as you could imagine with the FDA, and not to expect too much of them, and not for them not to get too much from us. I've had some applications to FDA that have not. I don't ever have had one that was rejected.
Yeah,
I've had some that were delayed. They didn't come out on time, but they were. I don't think anything has been rejected.
Yeah, if you bring it to them early and you're open with them and take their advice, sometimes you win the day. With FDA.
Exactly. Oh, you better be honest with them. But oh yes, for sure. And get their advice, as you just outlined, sooner rather than later.
So, John, you've been pretty successful at raising capital for your ventures. Do you have any tips on how to go about that, other than having a good idea and some preliminary data?
Well, I would be honest with you. Raising capital now is the hardest thing that I do. There was a time when it was the easiest. You just have a good idea, you go out and people just throw money at it. Those days, that ship has sailed, as they say in the trade. So now that it's very, very difficult, I think, to raise capital, you have to really selectively target the investor base very carefully. And even those that are sophisticated and that do a lot of investing in this space may or may not have interest. So it's I wish that I had the secret potion for raising capital. I think inevitably you seek out more capital from the strategics early on, trying to get some kind of a collaboration with Abbott or Asahi or Barstrom. I've had more success with Abbott than anyone else, but and I think it's been mutually beneficial for Abbott. I do have to put one little plug. I just thought they just have sold their 7 millionth Perclose device last year. I thought maybe they would sell 100 of those at some point, and now they're up to 7 million. But that's the other thing I wanted to say. You may not be able to predict not only the outcome for how you're going to get this thing funded. You may not predict the clinical value that's ultimately going to be derived. It may far exceed your expectations. I'm quoted in some San Mateo Times article saying there might be 500 patients annually that will have percutaneous coronary interventions. That might be a stretch. Maybe it's 400 annually. So not a real visionary when it comes to forecast models and things on those lines. And raising money is kind of one of those things. You got the usual NEAs and the I'm blocking the name. Institutional Venture Partners. Can't think of their acronym right now, but anyway, you got the usual venture groups, but they've moved away from new startups. And so it's more complex than it used to be. I wish I knew the answer. I don't, to be honest with you. I just have to call as many people as you can and try to give an honest statement about what you have.
Dr. Simpson, what do you recommend for students who are interested in innovation? Sometimes you see who's the innovator, how a person is innovate, and I hear sometimes that the innovators usually they have their life exposure provide the raw material to them, and when they see unmet need, their brain redesign that exposures and they can't find a solution. And some other, you read so many things about the innovators. I mean, how do you describe for the student the innovators and how should they trust themselves? Everyone can be innovators, or is that something just genetic?
Well, I promise you, everyone can do it. If I could do it, then anyone could do it, guaranteed. That I grew up raising Shetland ponies in Lubbock, Texas. So that I did not go. I flunked out of Texas Tech. Well, I didn't really flunk out. I just on the edge before I transferred to Ohio State. I've never been to an Ivy League school, so I didn't do anything correctly. This is just about how you. I just persevere. Somebody said, you're just not smart enough to stop. If you're any smarter, you would have sort of given up on this by now. And I think it's about, believing in yourself, believing, persevering. I absolutely want to be a team player. Nobody does this stuff by themselves. That you have to find really, really good engineers, really good fellow clinicians to work with.
Oops.
We lost our speaker.
Most likely his power, yeah.
I appreciated that shout out that John gave me, because he just said that if you were smart enough, you would give up. And that's what I told him to do when he came to me with this idea in the beginning. So that means I'm very smart, right? but not as successful in some things. Bill, I don't know if we'll get John back or not. And
yeah, I think we can go ahead. Tara, did you say they would like to introduce something about the program? Or what do you have next?
Oh, sure. I mean, basically, we're just going to continue the Q&A in case the audience has any questions. They can submit questions or thoughts. And I think this is a really open forum. It doesn't have to be a question per se. If you have any of your own experiences that you want to share in the Q&A, we'd be happy to moderate them. Professor Karen Goff's doing that. Maybe at this pause, why questions might come in, Professor Goff, do you want to describe what you do in the Regulatory Science Colloquium? And it's a year-round program that we offer where we try to topics such as this one that we did today, and I think an emerging issue in regulatory science, that I'll have Professor Goff describe what we're doing in this course in the program.
Sure, it's really a cool, unique course. It's 1 credit hour and it's a speaker series that all the speakers look at topics related to the intersection of science and regulation as it pertains to health. So we start off with some pretty basic stuff like, human subjects research, and then we go into a lot of specific topics like the one we have today. And, it's a colloquium style course, so we really encourage critical thinking and discussions, and the students seem to really love it. It's really intellectually engaging.
Yep, and about a third of our students are future lawyers, but the rest are from pharmacy, the medical school, public health, and also our School of Business, and very prominently biomedical engineering. So it's a model that did not exist when I was in law school that I think is a wonderful development. And look, pleasure to have you join us back. So I'll let you finish.
So stupid. This so dumb, so stupid. I just couldn't. You think I could plug my charger into my computer, don't you think, at this phase of my life?
Yeah, we're talking about the team, how important the team and everyone.
Yeah. Well, if the team was doing the job correctly, they would have plugged this thing in.
That's correct.
While you were gone, John, Dr. Goff was talking about the interesting question of how you go about in that phase 1. How do you decide that you've got enough safety data in the preclinicals that you can go ahead and ask people to volunteer, as Spencer or John?
I have an opinion, but let's ask Spencer first.
You're muted, Spencer.
Spencer, you're muted. You're still muted.
This is the quietest I've ever heard Spencer.
Yeah. So you're asking how do you decide when to go into human trials. I mean, you got a lot to do before you do that ordinarily. I mean, it's not like the days, John. It's not like the days when you and Andreas were, you stuck it in a baboon. Andreas put in a dog. I mean, the next time from the dog it got in anything was in a human. It got in a human in the operating room, but this day and time, how if you had a device that had this much risk to to the use of it,
The balloon, if you had a balloon, just if you're trying to get the balloon approved today.
exactly,
For instance,
exactly.
balloon approved today.
I mean, I had Grunsey come over in 78, maybe it was 79, and speak to a group, and I had a surgeon named Francis Robachek be the antagonist. And Andreas presented like 50 cases he had done. And the surgeon took him on and just destroyed him. He just said, in any device, any drug, anything that only worked about 60% of the time and the rest of the time caused all these complications and people had to be rushed to emergency surgery. I said, this, we would never approve such a thing. Now we're worried about 7 people having a thrombosis in their venous sinuses, and we hold back a vaccination where 7 million people have benefited from it. So it's a whole different, this whole risk thing for getting into human trials. Where is it today? What if you had you've got some new ideas. What do you have to prove before you can start human trials?
Is that for me? Is that
safety? Yeah, that's for you because you're the one. You've got a new deal, a new company I'm aware of that you're trying to do this.
Mm-hmm. So I think I mean this is a really good question. It's kind of a key question. I think we would always start off in the peripheral vessels. So for the small devices, for example, a device that's about the size of a coronary artery, the vessels that are similarly sized to coronary arteries are below the knee vessels. And so we'd start off there. We tested and evaluated in that setting and just look at our success rates and the safety profile. The new device, in my mind, has a safety profile that's going to be off the charts because we can see what we're doing, unlike fluoroscopy, which gives you a good clue, but doesn't really tell you the answer. This says exactly what you're aimed at, literally within microns. So I think the safety profile of this particular device was going to be higher than anything else that I've ever worked on in the past. Now, it's got to be smaller, a little bit just flexible. It's got some other issues. But the safety profile is the absolute key to deciding when to go to patients, right? You want it to work, but for sure it has to be safe first. And that is if it's proven to be safe in peripheral vessels below the knee, if you try to get it at the coronaries, then that's a good starting place.
Yeah, for everybody who's not in clinical work, you recognize right away that if you poke a hole in the artery in the leg, it's a whole different story. If you close an artery in the leg, it doesn't cause a heart attack. If you do it in the heart or the brain, you've got a different thing. So many of these things can be used in, as John has done, used in peripheral vessel disease where the risk is not nearly as great, but sometimes the reward may be even greater. I've heard you, John, pontificate on the value of opening up somebody's leg artery and saving them from an amputation. And the patient appreciates that more than fixing his heart, which he doesn't really notice.
Oh, so much more. I mean, Doc, thanks. My chest pain's a lot better. You want to go have a beer? You save somebody from getting their leg cut off, and it's like a game. It's just like, no, totally different than like you say, the patients can't really see their heart's not getting the blood supply that it needs. They feel it, but it is fascinating. We're really fortunate. And I would say this to all the students. I don't care if it's medical or law or I don't know, public health, you name it, whatever it is. We're so fortunate to live in this era when there are these opportunities that are afforded to us. And by the way, too, men and women, and I noticed that the women outnumber the men on this slide, or it's pretty close number, I guess. Also that
Because they're lawyers.
Oh, I see. Maybe that's a problem. Maybe that's a truth. But Ann did say, I object to what she said. My daughter saw some of my slides, and she said that the quote about there are men that go off on a path all on their own. That's not true. There are men and women that go off on a path all based on just on their own vision. So I want to correct that slide.
Yeah,
we have a question in the chat. Sorry, go ahead. Are you going to say something? This question is for Dr. Simpson. What does safe mean? I know it depends, but they'd like to hear your perspective on that.
Well, yeah, so safe means no adverse event. So adverse events come in several different categories, in particular in the coronary space. You could have a heart attack, you could have a stroke, you can have other events, but the major cardiac events by the FDA standards are pretty well defined. So that in the event that you would have one of those, the question is, as Spencer's just outlined, suppose that we say there's this so-called composite index, safety index, where you add all of things together. And if it's at a certain number, then it's approvable. If it's not at a certain number, it's not approvable. Is kind of one way to think about it. If you want to know what my own personal profile is, that is, it has to, in the coronary arteries, you have to be able to believe that's going to work every time. And that's I know that's a high number, but you've got to believe that, and you've got to have something that will give you the chance to do that. And we look at right now with balloons and stents. Balloons are not so predictable, but the stents are really predictable. So that's why they're viewed to be so important in the safety profile.
And this safety, it goes on after post-market, after you got a product and it's out there and it's been used in a lot of people. And John will remember that one time, one of his inventions, which is called the Anthrectomy device, that actually cut out a part of the plaque from the inside of the artery, was used. It started the company called DVI. Well, I was one of the principal investigators of this kind of device, and I used one day, and using it, the whole nose cone, the distal part of it, fell off in the coronary artery. And I called the company, and the company shut it down. Now, I found out some others had come off, and John's company had to redo things. But the next thing I saw was a slide that John had put together that was the gorilla in the Samsonite luggage ad where he's throwing stuff around, and he had my head stuck on the gorilla. So
this is an exaggeration, but that's okay.
It's absolutely worth exaggerating because there are things.
By the way, we did have some devices come apart. We did.
Yes, I know, and I one of them was mine. Yeah. But it was and at the same time, a device I was working with the USCI company, it broke, the end off broke the same day, and that company got shut down completely for a while. So safety continues. I mean, safety is something that's out there, just like this vaccination stuff. They do surveillance, but it's. And John says you never have these events, but you're always going to have something. I mean, somewhere along the way, 737 is going to crash, and it's not going to be good. Somewhere this vaccine is going to cause a side effect. But so safety is not 100% ever.
I have a great experience. I was doing the very first patient with a new agent that you injected intravenously, and it enabled you to outline all the chambers of the heart when you're doing ultrasound. And this was a brand new agent, very first patient. And after the study was done, I gave him a voucher to go down to the cafeteria, get something to eat. And about a half hour later, I got paged that he collapsed and he had a heart attack. And we rushed him up to the cath lab and opened up the vessel. But there we were, the very first patient, and stopped and went back and thought about it and you know, wrung our hands a little bit. And that was a tough one, but we went ahead and did the second patient. The bottom line is that that's now a product. And if we had stopped at that first patient, you know, we never would have made it across the goal line. Wasn't my product, but I was just the investigator.
These are examples of why we need your law school and your lawyers. we in medicine need.
It is 1 o'clock, so I want to be respectful of everybody's time. Thank you for the example. This was, I have so many questions. I could go on for a long time. We really appreciate you all being here. And for the folks who are listening to the webinar, the recording will be going out. I believe if you registered, it should go to your email. And check out both ISCTR and Arizona Health Law on social media, including Twitter and LinkedIn. And in addition to the regulatory science course, we do talks like this in our series, usually 2 or 3 per semester. So we'd love to keep in touch. And again, thank you so much to all of our speakers. I would love to continue. But yeah, have a wonderful rest of everyone's day.
Thanks.
Thank
This
you.
is truly important for us. We thank you all for this starting up. This is a great summer course with Tara Sklar between the ISCTR and the University of Arizona. So that's a very special session. Thank you so much, Dr. Simpson, for sharing all your expertise, and thank you so much for all your contribution to the science and to the patient. Also like to thank Dr. King and DeMaria for being panels today and bringing the session to life. So thank you so much, and we'll send the recording to everyone registered. We have over 100 registered and 60 attendee today. The one who did not attend also will receive the recording. So that's a recorded session.
Excellent. Fantastic. I enjoyed it. I thought it was good. If I ever do it again, I'll plug in my computer next time.
All right. Goodbye, everybody.
Fantastic, John. Thank you so much.
Thank you so
Okay.
much.